Good afternoon, folks. Thank you so much for joining us in our glorious bioinformatics cave. I hope you had a good lunch, and I hope you're ready for a couple of enthusiastic minutes of a little bit of microbial genomics. So everyone looking forward to this? Yay, awesome. That's what we like, a little bit of team engagement. So anyhow, I'm Stephen, and I'm looking after our Epitome Labs product. So could I have the kind of the gridiron screen? Perfect. So all I'm going to do briefly is I'm just going to talk a little bit about what is new from the land of bioinformatics. So hopefully some of you are familiar with our existing Epitome Labs software. We've been developing Epitome Labs for the last couple of years, and we've got a robust collection of workflows. So Natalia here is showing the collection of workflows that we have available on the system. These are pre-installed workflows that demonstrate best practices for solving contemporary problems in data analysis. We're looking at a variety of different scales of biology. We're looking from small genomes, such as SARS-CoV-2 or monkeypox influenza, through to human-scale genomics, and we're looking even at pairs of genomes with our recent somatic variation workflow. So Epitome Labs is a good starting place if you're looking for some inspiration as to how we could or would solve problems in bioinformatics. Our new product, which we're going to be talking about increasingly, we've got a register your interest QR code thing over there, enables the same familiar analyses that you've been using on device, on your laptop, on your gridiron, but you can run the data analysis in the cloud. This means that you can be a little bit more ambitious in the way that you run your studies. You do not have to run data sets that are restricted or sort of appropriate for your own compute resource. What you do is you just specify that you would like to work with the cloud, you build a little bit of infrastructure, you submit your jobs to the cloud. The jobs run in your own environment, it's clean, no data is returned, no data is retained, lots of data is returned. Hey, everyone was listening, I like that. Anyhow, you get some glorious results back, you get some beautiful reports, and lots of data that builds confidence. Anyhow, let's have a look at a couple of examples. So Natalia here is going to speak next, and she's going to talk a little bit about our metagenomics workflow. So Natalia, please.
So hi, everybody. I'm going to show the metagenomics workflow, which is our pipeline to analyze metagenomic data. So as you probably know, analyzing metagenomic data is a challenge because there are too many possibilities. So that's why we focused on elucidating the taxonomic composition of the samples. And the best way to run this pipeline is through this fantastic app. So instead of just me talking, and like a picture is worth 1,000 words, let's take a look and see how we can run the workflow here. So first thing, and most important, is setting the path to our samples. So in this case, we are the samples were taken from different biofilms collected from different surfaces for different factories that work with chicken and pork products. So once we have set up the path, then we are ready to launch the workflow, but there are other options. We can switch between two different methods to perform the taxonomic assignments. We can switch between Kraken2 or Minimap, and also we offer two different possibilities about databases. You can use our default databases, but also you are able to use your own custom databases. Selecting the database is very important to get a good results. So in this case, we run the pipeline using our Kraken2 database that we've made personally with some species and also with some chordates. And now we are ready to launch the workflow. And there are other options also that you can also customize. And then let's take a look about the report that you will get. We have generated a report earlier today, so we are going to speak a little bit about it. Basically, in the report, there are different sections. The first thing that you've seen will be some statistics about the length and the quality, just to make sure that nothing weird has happened, and you can trust the results that you get. So then, there are some fantastic Sankey plots so that you can explore the different taxa that is present there. You can switch between samples, also selecting different ranks, filter by abundance, and extracting your first conclusions. And then it's also possible to make some comparisons among samples in the bar plot. So you can see, for instance, to follow a time series, or just comparing between groups of samples if you need it, also to detect if you got some unexpected results. That's it. But as we know that you want to do more complicated analysis, just feel free to export the tables and play with them later. So we have the quick export button. Finally, we've included some statistics, some indices about diversity and also about richness, and some species richness curves that can be helpful to know what to do and what decisions made in the moment. Because yes, it's possible to run the pipeline in real time. So while the sequencer is producing data, you will be able to check the results, see how it's going, and decide to stop or not stop the pipeline according to your necessities. So basically, from this workflow, you just have to remember that you can run it on the device, on the cloud, in real time, and you get some results to know what's happening there, to find out what are in your sample, what things you can find there, microbial. And so now, I'm just going to let Matt to talk about bacterial genomes.
Hi. Thanks, Natalia. Thanks, Stephen. Hello, everybody. I'm Matt. I'm in the customer workflows team. I'm a bioinformatician, and I'm here to tell you a bit more about one of our workflows today, workflow bacterial genome. It's perhaps the logical next step following on from your metagenomics assay. Perhaps you've isolated a bacteria of interest, you've grown it up, and perhaps you now want to know what genes are present, what other features are there in this organism. You've done nanopore sequencing, and now you can do your analysis. So our workflow bacterial genomes allows you to do a reference-guided approach or a de novo assembly approach. Stephen's showing these features here now. So if you want to do a reference-guided approach, obviously you have to specify your FASTA file. We've also included a few new options in bacterial genomes that we're launching in this great Epitome Labs release for doing sequencing of isolates. So here we wanted to enable users to do antimicrobial resistance calling. So we've included ResFinder in the workflow. If you want to run ResFinder, like all our workflows, it's very easy to select these options. Stephen's here showing you, if you already know the species, you can enter the species here, and that will give you point mutation-based antimicrobial resistance calling. If you don't know the species, we'll still report on acquired resistance. As always, you can go into extra options to customize things like CPU cores, all that good stuff, and launch the workflow. So like all our workflows, we produce really easy to read HTML reports that you can share with collaborators. And we ran this workflow earlier, and Stephen's just going to show this to you now. So this is a summary report. Like Natalia explained, we like to produce quality statistic information about your run so that you can tell if something might have gone wrong. Things like read quality, read length, and all that good stuff for each of the samples you've got on your run. The next table here shows you a little bit about your assembly, so how much coverage you've got of your config, how well your sequencing experiment worked, also if you've got any circular contigs in there, so if there's any plasmids present. We also show you plots of things like genome coverage. If you've chosen a reference-guided approach, we'll show you how many reads you've managed to achieve across your genome. The next part of this workflow, it annotates these assemblies with Procker to give you information on what features or genes might be present in your assembly. So the new feature we've added recently is antimicrobial resistance. So for this sample, for example, we don't know the species and there are only acquired resistance displayed. But for this sample, we know it's Mycobacterium tuberculosis, and so ResFinder has gone away and given us all the point mutations in this sample that might contribute to antimicrobial resistance in this isolate. You can run this workflow locally, really easily on any of our devices on your laptop. It's really low resource. You can take it into the field, run Epitome Labs on a laptop and get great reports. And it produces all of the files that you would need to do phylogenetic analysis or anything you want to do with a FASTA file after you've done the sequencing. And of course now, the great thing is you can run this in the cloud with one click. Thanks a lot for listening.